Welcome to Art Medicos. In this section, we are discussing the second part of our physiology, basically the general physiology. In general physiology, we are discussing the topics that link all the topics of the physiology. It means maybe the systemic physiology, the digestive, thorax, that is respiratory, circulatory, or the neuro and neurophysiology. We all the physiology is connected with these topics. So the general topics we are going to discuss here. The first we discuss about the cell organelles and what are their function of their parts and the different types of proteins and their receptors. Now in this chapter we are going to see what are the secondary messengers, what are the receptors, what are the main enzymes they are actioning in that area, or what are the hormones that regulating the activities in the general physiology here in this topic. In the general physiology part 2, we are going to basically discuss about the cellular messenger or the secondary messenger and the receptors. What are the receptors which are located? They are the extracellular or the intracellular receptors. The extracellular receptors are the receptors which are present outside the cells on the cell membrane. Suppose that this is the cell and on the cell there is a cell membrane. These receptors are present. The receptors are present on the cell membrane. Some receptors are present inside the cell membrane. That's why they are called extracellular receptors and the intracellular receptors, like this. The extracellular receptors are basically present outside the cell and intracellular inside the cell. Basically in intracellular, two types of receptor. One is the cytoplasmic and one is nuclear. It means what? Inside the intracellular receptors, there are basically two types. The one is in cytoplasm, inside the cell cytoplasm, and inside the nucleus, that is nuclear receptor and the cytoplasmic receptor. And what is extracellular receptors are what? They are located on their circumference, their cell membrane of that cell. These are the two types of basically receptors. Here we are going to see about on the basis of the entrance of these molecules, what are the types of hormones. Suppose that this is the cell and some molecules, this is the some chemical substance, they are going to bind on the surface of the cell and they enters inside. They are binding on the surface and they enters inside the cell. They crosses the cell membrane. Suppose that this is the cell and this is the chemical substance. They are going to bind to the cell and after that they enters inside the cell. It means they are entered inside the cell. They crosses the cell membrane. But in another word we can say, suppose that these chemical substances are coming and bind to it, but they don't transfer inside it. It means they are not crossing the cell membrane. They are waiting on the surface of the cell. It means these are called as extracellular receptors. They are coming and binding to the surface receptors, that is cellular, extracellular receptors. What are the chemical substances? They moving inside the cells and they binding to the receptor is intracellular chemicals, intracellular hormones. So these are the two basically types of the hormones, that is extracellular and intracellular. We are going to see on the basis of the introduction part. We can see here the hormones can be classified into two types basically. What is on the which crosses the cell membrane or do not crosses the what? Cell membrane. Either they crosses the cell membrane or either they do not crosses the cell membrane. The receptors are located inside the cell. When the receptors are located inside the cells, when they crosses the cell membrane, when the any part enters inside the cell, then it will crosses the cell membrane. It means they are going inside the cell. And this type of action is happen directly inside the cells, directly influencing the cellular function. When the any chemical substance they hitting on the surface of the cell and after that they moving inside the cells, after the crossing the cell membrane, they functioning, they influencing the cellular function directly because they entered inside the cell directly. Suppose that in second case we can see, suppose that this is the cell and this is the chemical substance. They're going to bind on the surface but they do not enter inside the cell directly. They are binding on the surface and they activating something inside the cell. That is called as a secondary messenger. They are activating something inside the cell. Suppose that this is the cell and this is the chemical substance. This is going to bind on the surface of this cell. After that, what happened? Inside the cell, a special chemical is activated due to their attachment on the surface of this chemical. Inside the cell, there is a second messenger is activated or the second chemical is activated. That one is called a secondary messenger. And they are activated in when they do not process the cell membrane. We can see here they act on the receptors which is present where on the cell membrane and they released their action into the secondary messenger. After that, what happened? The secondary messenger, they influencing the cellular function. The first, what happened in the extracellular intercellular activity, they are binding, they are moving inside the cell, and they are showing directly their effect. In next one we can see they are binding to the receptor. After that they activating the some another chemicals, that is secondary messengers, and then their effect will be shown on the cell. We can see what is the classification of the chemicals, how they classified. The classification, what are the chemicals, what we are talking now. Suppose that this is the cell and this is the chemical, they are binding and hitting and they are moving inside the cell. What is happened? What are the chemicals which are binding here? The chemicals are hormones basically, they are proteinaceous in nature. These are the hormones we are going to classify, how they are formed on the basis of their formation. What are their nature of these hormones? They may be proteinaceous, they may be amino acid derivatives, they may be peptides, like this. We have to discuss about it. So now we are going to see first classification of hormone, in which we are going to discuss about the amino acid derivative. Some of the hormones are derived from the amino acids. Amino acids are basically, we are going to see single amino acids. They are the just a single amino acid is drives several hormones. What are the amino acid we are going to discuss here? They produce some hormones. Basically there are the three we are going to discuss now. One is tyrosine, tryptophan or arginine, TTA. We can see tyrosine, tryptophan and arginine are the three amino acids. They producing several hormones. What are the hormones produced by the tyrosine? T3 and T4, triiodothyronine and thyroxine. These are the two thyroid hormones that is produced by the tyrosine amino acid. Next one, catecholamines. Catecholamines secreted from your adrenal gland. Adrenal gland, which part? Cortex or medulla? Medulla. So these are secreted by the cortex medulla part. And what are the catecholamines? Catecholamines are the group of some hormones, and there is included three hormones basically: epinephrine, norepinephrine and the dopamine. These are the three hormones group, that is epinephrine, norepinephrine and the dopamine. These are combinedly called as catecholamines, and they are derived from which amino acid? That is tyrosine. Next one, tryptophan. Tryptophan is forming two hormones, that is 5-HT, that is 5-hydroxytryptamine and the melatonin. These are the two hormones that is synthesizes from the amino acid, that is tryptophan. Tryptophan is synthesized 5-HT, 5-hydroxytryptophan. This is also called as serotonin. Serotonin, another name of serotonin is 5-HT hydroxytryptophan, and melatonin is also derived from the tryptophan. Next one is arginine. Arginine is deriving a special type of neurotransmitter or the hormones. That one is called as nitric oxide. The nitric oxide, NO is synthesized from which amino acid? That is arginine. These are the three amino acids which forming these hormones. We have to memorize it, which amino acid forming the which type of hormones and which amino acid forming the which hormones. Basically we are going to see next one, proteins or peptide hormones. Basically these are the group of amino acids. Suppose that the one amino acid we are going to discuss in first part, we are going to see the single amino acid is producing something. The single amino acid is producing some hormones. These are called as the their amino acid secretion. Will suppose that the arginine, tryptophan or the tyrosine, what are they? Now we are going to discuss about the protein, the group of amino acid. They are the 2, 5, 9, 10 like this. They are forming a group, and after that they are producing the hormones. They are the many amino acids are combined, and they to form peptide bond in between them, and they are forming proteinaceous structure. So these are the amino acids. They are the many amino acids which combined to forming the peptide or the protein hormones. They are forming the protein hormones, as I told you. So there the 51 amino acid is combined together and they are forming a hormone. That one is called as insulin. So the insulin is synthesized from the 51 amino acids combined together, and they are forming insulin. That's why the same we can see 84 amino acids are coming together and they are forming a special type of hormone that is called as parathormone. Or the parathormone, we can see these hormones do not crosses the cell membrane. What does it mean? Their receptors are located where? On the cell surface, on the cell membrane. That's why they are not crossing the cellular barrier. And these are called as extracellular receptors, where they acted on it. Extracellular receptors, when they are not able to crossing the cell membrane, it means for them, for these hormones, for these chemicals, the receptors are located on the cell surface here. Suppose that this is the cell, on the cell surface these receptors are located. These chemicals, the parathormone or the insulin, these are binding to it and showing their actions by activating the secondary messenger. We can see now the third part is the cholesterol derivative. We can see cholesterol derivative. What are the chemical substances which is derived from the cholesterol? Suppose that this cholesterol is crossing or not. This is crossing the cell membrane, yes, and they acting on the intracellular receptors. It means they are acting on intracellular receptors. We discuss in basically in two parts, cytoplasmic and nuclear receptors. They both are the intracellular. They are located inside the cells. Many steroid hormones are the intracellular receptor section, and they are derived from the all the, basically we can say like this, the most of the steroid hormones are acting intracellularly inside the cells, in cytoplasm, either in the nuclei. And we can see steroid hormones. What are the steroid hormones? We can going to write a list of names: aldosterone, cortisol, estrogen, progesterone, and testosterone. So what are the main component of it? That is estrone, androsterone, progesterone, estrogen, or the testosterone. So these are the names basically, or the cortisol, which are derived from the cholesterol. These all are the derived from where? Cholesterol derivatives. These all are the cholesterol derivative hormones. Now next one we are going to discuss about the vitamins. What are the vitamins? They acting as a chemical substances. These are the vitamin A and vitamin D. They acting as a hormones. Vitamin A is called as retinol acetate or the retinic acid, and the vitamin D is, we can say like this, these are the two fat soluble vitamins. Vitamin A and D is the fat soluble. What are the other fat soluble vitamins? A, D, E, K. These all are the fat soluble vitamins. That is, they crossing the cell membrane or not? They crossing the cell membrane. But basically, what are the vitamins which producing the hormones? Only A and D, and they crossing the cell membrane. Yes, it means they are showing their action inside the cell. That is intracellular receptors. They acting on the intracellular receptors. Once they crossing the cell membrane, it means they acting on the intracellular receptors. Now next one we are going to see the receptors. In first part we discuss about the what are the chemical substances from where they synthesizes. In second part we are going to see the receptor part, where these hormones or the chemical substances are acting on the cell. Suppose that this is the cell. On the cell there is a receptors where these chemical substances, what we discuss in first part, they are going to bind on the receptors. So what are the receptors is located on the cell? What is the different types of receptors? There is two types of receptors as we discussed from the top. They are the basically we can see extracellular and intracellular receptors. Intracellular I already told you, that is cytoplasmic and nuclear receptors. And in extracellular receptors there are basically three. GPCR, Gprotein coupled receptor is the first one. Second one is TKR, that is tyrosine kinase receptor, and the JKR, Janus kinase receptor, or the JAK-STAT is the another name of this receptor, JAK-STAT. So we can see like this, the receptors are basically divided into two parts, extra and intra. The extracellular receptors are located on the cell surface. They are the three. Gprotein coupled receptor first, second one is Janus kinase receptors, and third one is tyrosine kinase receptors. These all are located on the cell surface. What are the inside cell? Cytoplasmic and nuclear. This is the classification of the receptors. Now we are going to discuss one by one. The Gprotein coupled receptors, you can also go in the pharmacology section and you can learn from there. Receptor topic is separated from there. We can also discuss there everything in detail. Now we are going to also discuss in detail, but little bit smaller, not that much larger or that much detail about the receptors here. We can also you can also go there and study. Now we can see here Gprotein coupled receptor. This Gprotein coupled receptor, or they are also in short form, they are called as GPCR. Their another name is 7-pass receptor, serpentine receptor, transmembrane receptor, or they are also called as heptahelical receptor. So these are the names of these receptor because they are passing the cell membrane 7 times. We can see like this: 1, 2, 3, 4, 5, 6, 7. That's why they are called as serpentine receptor, 7-pass receptor, heptahelical receptor, transmembrane receptor, because they are located inside the membrane of the cell. That is transmembrane. So these are the transmembrane protein receptor. This is also called as 7 transmembrane receptor, serpentine receptor, or they are located where? On the cell membrane. That's why the cell membrane receptors. there are the three subunits of these receptors, alpha, beta and gamma. There are the three subunits of these receptors, alpha, beta and gamma. We have to memorize it. What are the three subunits of the G protein coupled receptors, alpha, beta and gamma? We can see this alpha, beta and gamma is having what type of activity is it? They showing activities is GTPase. It means they having enzymatic activities GTPase. They activating the GTP activity. We can see in the next step, we can see the GPCR activation means G protein coupled receptors. They are located on the cell surface. They are showing their activity how. Suppose that this is the cell. On the cell surface, they just suppose that this one is the GPCR receptors, and how they gets activated. We can see here how many subunits are here. 3 alpha, beta and gamma subunits. We can see, suppose that this is the cell and this is the receptors, and here is the alpha, beta and gamma. We can see this is the receptor. Inside it there is alpha, beta and gamma subunits are located inside it. What happened? They gets separated when the GPCR channel, when the receptors are, some chemicals are coming and bind to it, they gets activated, and alpha, beta, gamma subunits are located inside this. After they gets separated, they separated from beta and gamma segments alone. The beta and gamma moving aside, the beta and gamma alone, and the alpha is alone. They are moving like this, and the GTPase activity is shown. The GTPase is gets activated. What is the GTPase activity? There is a conversion of GTP is hydrolyzed. How the GTP is hydrolyzed? The GDP is gaining 1-phosphate. We can see here the hydrolysis of GTP and the production of energy and leads to the GTPase. Due to them, there is a downstream action of these hormones. These hormones are gets activated. We are discuss very short about these hormones here. Basically, I am trying to say you, this is the receptor GPCR, in which 3 alpha, beta and gamma subunits. The alpha is get separated from the beta and gamma when there are some chemicals are binding to these receptors, and this activation of alpha and beta gamma subsegments, they leads to the several changes inside the cells. That is called as phosphorylation and dephosphorylation inside the happen. Basically, there is a phosphorylation of the proteins or the components of the inside the cells. Their cellular components is having phosphorylated, and the synthesis of the protein and the different parts, different types of channels will be formed there, and the activity is happened. We can see in this when the hormones binds to GPCR. When the hormone, suppose that extracellular receptor, GPCR is extracellular, there is alpha, beta, gamma subunits. This is the primary messenger. Primary messenger is what? That is hormones. That chemical is primary messenger, and secondary messenger is what? We didn't discuss till now. What is secondary messenger? Suppose that this is the cell membrane. We can see here. And on the cell membrane, what type of receptors are located? For binding the hormones, that is GPCR. And here is the alpha, beta and gamma subunits. And they produce secondary messengers. What is secondary messenger? They transferring the information from outside to inside the cell. It means what? They transferring information from here to here, from first to second. This is the transferring of information through them. That is the 7-pass receptor or the GPCR. We can see the second messenger or the secondary messenger into the cell is exclusively seen in the case of GPCR receptors. First of all, we are going to discuss about the extracellular receptor. In extracellular receptor, basically we are going to discuss now is the GPCR. In the GPCR, there is a 3 segment, alpha, beta and gamma. In the alpha segments or beta gamma segments is separated when they are gets hydrolyzed or they gets activated. What happened in the secondary messenger digit? They are the basically activated in the GPCR only, most of them time. What is the secondary messenger? What is secondary messenger? This is the primary, the chemical binding to it inside the alpha, beta, gamma subunits. They activated and they produce a secondary messenger. They transferring the information from top to inside the cell to the secondary messenger. And the secondary messenger are 3 in number. They are, first one is cyclic AMP, second one is calcium plus 2, and third one is cyclic GMP. The cyclic AMP, C stands for cyclic AMP, cyclic GMP, and calcium plus 2. AMP, what is it? Adenosine monophosphate and guanosine monophosphate. We can see, suppose that The patient is having calcium plus 2 is a secondary messenger. From where they come? We can see, suppose that this is the cell. On the cell, this is the cell membrane. Suppose that we are going to larger it in the specific region. There is a phospholipase enzyme is located on it. They gets digest this cellular membrane by the phospholipases, and they produces two chemical messengers, basically IP3 and DAG. After that, they going to action on the sarcoplasmic reticulum, and the release of calcium plus 2 is happened here. The release of calcium plus 2 is happened from the sarcoplasmic reticulum of the cell, or basically the muscle cells, and they activated basically IP3 and DAG by the way from the cell membrane. And cell membrane is digested by what? Phospholipases enzymes. So we can see here phospholipase C is basically the enzyme basically action on the cell membrane, and cell membrane is made up of the phospholipid layers, that is phospholipids. These phospholipids are gets digested and production of IP3 inositol triphosphate and diacylglycerol DAG, and after that they acting on the sarcoplasmic reticulum, and they release of calcium from the sarcoplasmic reticulum, because the sarcoplasmic reticulum is the endoplasmic reticulum, and they are the store of calcium in our cells, in the muscle cells basically, and they activating alpha Q, or the quiescent is gets activated. We have to memorize it. Alpha quiescent is activated. What is alpha quiescent? The three subunits of what? GPCR is having how many segments? How many subunits? Alpha, beta and gamma. I already told you that alpha segments is separated and beta gamma segments is separated. The alpha is maybe alpha the S stimulatory, alpha inhibitory, maybe alpha quiescent, maybe alpha not, or the many more, but we have to discuss only alpha stimulatory, inhibitory and quiescent. We can see here alpha stimulatory. When the alpha stimulatory is gets activated, then what happen? Inside the cells, the cyclic AMP concentration will be increased. We are going to discuss here the secondary messenger, and we discuss here secondary messenger 3, cyclic AMP, calcium plus 2 and cyclic GMP. These are the three secondary messengers. The cyclic AMP gets activated when a special type of enzyme is acting, that is called as adenylyl cyclase. Adenylyl cyclase. The calcium plus 2 gets activated when a special type of enzyme is acting, that is phospholipase C, as we know from the above. And cyclic GMP gets activated when the guanylyl cyclase enzyme is activated. Guanylyl cyclase. So these are the three enzymes from where the cyclic AMP, cyclic GMP and calcium plus 2 are gets activated in the form of secondary messenger. These all are located in the cell membrane of the any cell. Suppose that this is the cell membrane. They are located somewhere in the cell membrane, and they gets activated inside the cell. And inside the cells, what is activated? Secondary messenger. What are the receptors on which these chemicals are binding? They are called as first or primary receptor. First messenger or primary messenger. So this is how we have to understand these concepts. What are the hormones they are producing? What secondary messengers? We have to memorize it. What are the secondary messengers do you mean? We can see here. What does it mean? When the alpha stimulator gets activated, they acting on the adenylyl cyclase enzyme and stimulated, and they increases the cyclic AMP concentration. What is done by the cyclic AMP we discuss later, but we have to memorize now. The cyclic AMP gets activated. The cyclic AMP amount will increase when the adenylyl cyclase enzyme is increased. Next one, when the alpha inhibitory is activated. When alpha inhibitory means inhibition is activated from the GPCR, they gets acting on the adenylyl cyclase enzyme and they inhibit them. Inhibitory is inhibit and excitatory is positive. It means they stimulate them, and that decreases the cyclic AMP concentration. What does it mean? When the cyclic AMP concentration is decreased, it means cellular activity is depressed. It means they gets inhibited by something. So what the chemicals are binding to these receptors are inhibiting the cell. These are the inhibitory chemicals. And these inhibitory chemicals are called as inhibitory neurotransmitters. And when these chemicals are binding to the receptors and activating the cellular function, that's why they are called as the excitatory chemicals. And this is also called as excitatory neurotransmitters. So these neurotransmitters, they depending on the action of neurotransmitters. Suppose that the receptor is responsible for the GABA, that is gamma aminobutyric acid, or the glycine. These are the inhibitory neurotransmitters. When this binding to the receptors of the GABA A, GABA B, GABA C, these are the three receptors of the gamma aminobutyric acid. When they are binding to it, they are inhibiting the cell. That's why they are the inhibitory action. It means what happened inside the cell? Inside the cell, there is a decrease in amount of cyclic AMP. What happened when the excitatory neurotransmitter is binding? Suppose that the glutamate aspartate, these are the excitatory. When these are binding to the cellular activity, is increased. It means they are stimulating the cellular activity. What is increased in the secondary messenger increase in the cell? That is cyclic AMP is increased. So this is happened when the alpha stimulator is increases or the alpha inhibitor is activated. What happened in the calcium plus 2 phospholipase C? There is a alpha Q is gets activated when the calcium plus 2 is secondary messenger. What happened when the cyclic AMP is coming, cyclic GMP is coming? When the alpha T transduction means conversion, there is a phosphodiesterase enzyme which gets activated inside it, and they decreases the cyclic GMP. You have to memorize from what are the secondary messengers. They are the three, from where they coming, how they acting, and that's all for the secondary messenger. Now we are going to discuss one by one cyclic AMP. The cyclic AMP we are going to discuss here. Their main role is in water reabsorption and electrolyte secretion. What does it mean? Water reabsorption means we have to retain water in our body. We have to store water in our body. We don't want to lose water. We want to retain the water in our body. That's why we are going to do something with the cyclic AMP. When the cyclic AMP is activated with the function of water resorption, how they are going to act on it, we can see here on the collecting duct. Where the collecting duct is it? We can see here. Suppose that in the kidney, the functional unit of kidney is nephron, and the nephron is having several parts. Suppose that this is the nephron, and in the nephron we know this is the glomerulus, this is the Pachythy, this is the descending, or this is the ascending, and this is the commonly called loop of Henle, and this is Pachythy, and this collecting duct. We are going to discuss basically collecting duct part. In the collecting duct part, this cyclic AMP is acting, and there is an enzyme is binding, that is vasopressin. Another name of vasopressin is ADH, antidiuretic hormone, that is vasopressin. These enzymes are acting on the collecting duct region, and basically there is a receptors is present on the collecting duct, that is V2. V2 receptors of vasopressin is located. V2 receptors of the vasopressin is located on the collecting duct of the nephron of kidney, and where these receptors are acting, and when the vasopressin is come in from the blood circulation and binding to it, what happen inside it? This vasopressin is binds to the V2 receptors, and they increases the cyclic AMP concentration, the secondary messenger, and they forming the aquaporins. Aquaporins, what is aquaporin? Aquaporin is a protein. Aquaporin is a channel that is synthesized by the action of cyclic AMP. In the collecting duct region, we are going to synthesize. In this region, going to synthesize the some channels. The name of channels is aquaporins 2. These aquaporins 2 channel is synthesized by what? Cyclic AMP. After the vasopressin binding to it, they increasing the number of vasopressin, they increasing the number of aquaporins 2 channel. After it, what happened? The water is pumping inside more and more, and the water is get retained. So this is the main function of it, and the water is gets retained, or the reabsorption of water is happened. So the reabsorption of water is basically done by what? The reabsorption of water is done by the vasopressin binding to the V2 receptors on the collecting duct region, and they synthesizing the aquaporins 2 channels, and they leads to the water reabsorption. In electrolyte secretion, it is not the physiological function basically. When the toxins are increases in our body, suppose that the toxins may be the cholera, suppose that any disease is happened in the patient having, suppose that the cholera disease. In the cholera, what happened, there is in the intestinal part of the patient, in the intestine, suppose that the cholera toxin is present. This cholera toxin is leads to the NaCl, the sodium and chloride are lost through the stool part. That the sodium or chloride is lost from our body, that these are the electrolytes, and the sodium follows water. These all are going out from our body, and it leads to the electrolyte secretion. Electrolyte is going out or the moving out from the body. Next one we can see pertussis toxin. What happened in this? In the patient lungs is gets toxified and the electrolytes is lost in the form of sputum. Pertussis is basically caused by the whooping cough, the dog-like cough. They are the person is having cough and in which the patient having a sputum is coming, a lot of sputum. This sputum is containing several electrolytes and there is a loss of electrolytes in these patients also. In the first one is intestine. With the cholera patient, there is a cholera patient is having cholera toxin. It leads to the loss of NaCl through the stool. In this patient is having the sputum. Through the sputum, the loss of electrolytes. These are the not physiological, they are the pathological functions basically of the cyclic GMP, water reabsorption and electrolyte secretion. Next we can see calcium. Calcium are also the secondary messenger cyclic GMP. So now we are going to see calcium. Calcium plus 2 is having main function is contraction in the muscles and vasoconstriction in the vessels. Vasoconstriction. Suppose that this is the vessels. There is a this is circumference or the diameter that gets compressed. There is a vasoconstriction in the vessels. So there is a vasoconstriction in the vessels. We can see here, where the contraction is happened due to the calcium uterus, in the uterus, due to what are the conditions in which the contraction is happened due to calcium in the secondary messenger, that is normal labor condition in the child during the childbirth, and in the second it is due to the hormone oxytocin. Oxytocin is producing the normal labor during the contraction. Next one we can see vasoconstriction is done by the endothelin, angiotensin 2, norepinephrine and vasopressin. And the vasopressin V1, V2 is acting on the collecting duct and V1 is acting on the vasoconstriction. These are in the blood vessels, basically in the vessels, our vessels. The endothelin, first one, second one is angiotensin 2, third one is norepinephrine and the vasopressin V1. These all are the four is acting on the vasoconstriction for the vessels, and the contraction on the uterus region is done by the oxytocin. In the normal labor condition, that is done by the calcium plus 2. Now next one we can see cyclic GMP. In the cyclic GMP, they are doing two functions, relaxation and vasodilation. Where they do relaxation and vasodilation, basically they are done by the nitric acid or the natriuretic peptides. These are the two main chemicals that is responsible for the relaxation and vasodilation, and they are the atrial natriuretic peptides, brain natriuretic peptides, and CNP, that is the cholinergic peptides, that is the atrial natriuretic peptides, brain natriuretic peptides, and the cholinergic peptides. These are the three. Next one is other cellular membrane receptors. Other cell membrane receptors are basically, we are going to see extracellular receptors. Other we are going to discuss here the first one GPCR, in the second tyrosine and the Janus kinase. Tyrosine kinase receptors is basically for the insulin and growth factors. Growth factors, what are the growth factors? Insulin growth factors, or the we can see PDGF, or the many VEGF, that is vascular endothelial growth factors. There is many endothelial growth factors is located. These all are the growth factors of the insulin. They acting on the special type of receptor, that is Janus kinase or the tyrosine kinase receptor. What is the Janus kinase receptors? On the Janus kinase receptors is also called as a JAK-STAT receptor, and these receptors are responsible for the binding of twin hormones. The what are means twin hormones? The twin hormones means the two hormones which are located same, they are the acting same action, that is growth hormone and prolactin. These both hormones are called as twin hormone, and these hormones are acting on the same receptor, that is Janus kinase receptors or the erythropoietin. Erythropoietin is for the RBC formation. These are also located where? The gynokinase receptor. They are also acting on the Janus kinase receptor. Down regulation of these receptors. Down regulation of these receptors are the mainly in three ways: internalization, inactivation, and desensitization. This is all about the down regulation. We are going to discuss about later is about the down regulation. So we are going to basically take on three names: internalization, they must, or the destroyer of these receptors internally, and our next one is inactivation. The receptors get inactivated in the second step, and the third one is desensitization. The receptors become less responsive. They are the three main receptors. The receptors down regulation means the receptors get inactivated. How the receptors get inactivated? The three ways. After that we are going to see intracellular receptors. Already I told you the intracellular receptors are the receptors. Intracellular receptors are the basically located inside the cell. They are the cytoplasmic receptors, maybe cytoplasmic or maybe nuclear receptors. Cytoplasmic receptors, already I told you, they are located mineralocorticoid, glucocorticoid, androgen, and progesterone. These all are the four receptors are located inside the cytoplasm. And vitamin A, vitamin D, estrogen receptors, and T3, T4 thyroid hormone receptors, these all are located inside the nucleus. We have to memorize it. It is the most important MCQ questions from here. And these all are the receptors are present inside the cytoplasm, cytoplasmic receptors, and the hormone binds to the receptors inside the nucleus. This hormone binding forming the hormone receptor complex, and they move inside the nucleus and showing their action. This is all about the hormonal control regulation. This is all about the intracellular receptors and their activities. In this topic we are going to learn from this topic are the cellular messengers, what are the secondary messengers, what are the receptors we are going to see, and we are going to learn from here. So you can watch it and try to learn it from the what are the extracellular messengers and what are the intercellular receptors, and the extracellular receptors, what are the chemical messengers which are located inside the cells or outside the cells, what are the chemicals, what are the hormones, what are the single amino acid derivatives, hormones. We have to memorize everything from this topic. So we have to moving for the next topic is the third general physiology later. Now we are finishing this topic. We are going to discuss in next part, I think, again general physiology. So don't worry, you can memorize this. This is the most important topic for our upcoming lectures. So you can memorize it, and all the best from my side. Take care. Goodbye. Thank you.